It's a privilege to introduce our next speaker, Dr. Peter Wettstein. Peter's a cancer immunologist who works with many people at Mayo Clinic, and we're privileged in our laboratory to have him work with us on novel immunologic treatments for chordomas and other sarcoma tumors. And with that, Peter, I'm not going to say anything more about your talk because you're going to tell everybody. And thank you for coming today. This is the advance.
Okay, thanks. Well, I am honored to be able to speak with you today. And when Dr. Yazemski asked me if I would speak to this conference, I said yes before I knew what I was getting myself into. So I'm a trained immunogeneticist, and I've worked with human cancers. I've worked mostly in mouse models of transplantation and cancer immunology, but I went immediately to the computer. I saw a comment on your site from Soldano Ferroni, who is an old friend of mine, and I thought he said it very well. He had to look it up. So I thought I was in good company at that point. And you also made a great choice. And I applaud the foundation for the quality and extent of their knowledge on the web. My ability to come to speed is owed to you, and I thank you. When I told Mike I would talk, I had to think about, now, what am I going to say? Since he didn't also say that I'm emeritus and slowly my immunological knowledge is draining away. I thought you, especially knowing that this foundation and its members are well trained in immunology and other aspects of responses to cancer. So I didn't want to bore you with the entire immune system and how it works. What I want to talk to you about is another way to look at the battle against cancer that every individual has. And I sort of view everything in life as I look at it from microcosmic to macrocosmic, that societies and cities and towns and collections of humans down to humans and all their cellular components, all are operating under the same sorts of drives. Everybody wants to live, whether it's a cell or it's a society, and they develop ways to continue themselves or their successors. And what I wanted to do today, we'll get past this because I have nothing to disclose. If one looks at established chordoma therapy, you all folks all know this much better than I do. It's a relatively linear and sequential approach. And the question I'm asking, is there a logic for an integrated approach that's not sequential, and is there a potential role for immunotherapy in such an approach? And I'm drawing on the premise I'm making is that there is a role for concepts in conservation biology to teach us about interactions in the body. And if one looks at it that way, cancer is an invasive species. Okay? The human body is an ecosystem. It's essentially an island. It's comprised of multiple interacting and interdependent species. They all depend on each other. An invasive species can destroy an ecosystem. They can use all its resources. They're classic examples in conservation biology of overrunning an ecosystem. There's overpredation. And then there's the ripple effect through all the interdependent species. So can development of cancer treatments profit from analysis of ecosystem management? Can you take the lessons that are learned in that sort of discipline and apply them to cancer therapy? Humans excel at driving wild species to extinction. We're great at that. And it's achieved by reducing population sizes and destruction of habitats. So what you first do is you bring the size of the population down, and then you destroy the habitat. And if you think about it as I go through these, the problem with an individual dealing with cancer. Cancer is the population size. It's the population. And can we adapt methods for analyzing the effects of reduced population size and deleterious environmental factors for refining cancer therapies? That is, can we learn from all the people who develop population biology and how species, populations react to stressors in their environment? Can we learn from that and apply that to the battle against cancer? So population size has a tremendous effect on species survival. Population size and density dictates that large is better as long as the habitat can support it. And genetic diversity generally correlates with population size. So within a tumor, you have a number of cells, millions of cells, okay? They are genetically diverse because the mechanisms whereby they replicate their DNA and they sort their chromosomes are faulty. They're mutations and they're deletions. So the larger the population, the more diversity you have. And that is the more flexibility that population has to deal with stressors. And increased population size reduces the level of inbreeding, and that is how identical each cell is to another, and resulting homozygosity. So like an inbred mouse, like you folks are trying to produce, if it's a small population, you have a much greater probability that all the cells are going to be the same. And that if you treat them, they're all going to be treated the same. They'll respond the same. And it's important for a population to have a level of diversity that can enable it to deal with stochastic environmental changes. That is, random environment changes. Okay? So you have a tremendous number of environmental influences, abiotic factors, temperature, precipitation, toxins, level of predation, size and diversity of predator species. And as I go through this, to set the stage for how you deal with cancer, think of these same things in a cancer setting, in an individual. You have effects that you can level against it. There's predation, T cells, and essentially any factor that can negatively or positively affect species survival is an environmental influence. And then you have the interplay between population size and environmental influences. As I said, the population size, that is the tumor size, governs the ability of a species to adapt and evolve. Okay, the greater the genetic diversity, increases probability of mutations and insertions, deletions that can modify evolution of a species. So you can have single point mutations, which don't occur all that often. I mean, as little polymerases are making new DNA, they make mistakes, just like all of us make mistakes. And there are insertions and deletions. It's how the brachyury locus was duplicated. Okay? When the chromosomes lined up, they misaligned, and somebody got 2 copies. Somebody else lost it. But that occurs with a greater frequency than mutations. And it's big changes where whole genes can leave, or whole complexes of genes. And species survival in the face of changing environmental factors requires a sufficient number of survivors. And there's a concept called the Allais effect, which is positive density dependence. Whereas the population drops, so does the survival ability of an individual when it hits a certain threshold. So you bring it down low enough in the population size, you can bring down the ability of each individual to survive. And so you have multiple stressors of a population. And a population may adapt to a single stressor with a subset of individuals surviving. Okay? So if the chance of surviving is 1 in 10, and your population is 100, you'll have 10 survivors. Well, if your population is 1 million, you'll have a lot more survivors. And the extent of survival is dependent on the size and diversity of the population. The larger the population, the more diversity, genetic diversity, the better the population's chances of having individuals that can adapt. And survival in the face of multiple stressors is the product of the survival probabilities to the single stressors. So if you have one stress that's 1 in 100 survival and another that's 1 in 100, you apply them both. Survivors are only 1 in 10,000. So the addition of each stressor, each stress that you apply to the population, drives down the number of survivals exponentially. So how do you drive extinction? And these are well-known concepts in conservation biology, and they're well practiced by the human population. You reduce the population size. You apply multiple negative stressors while the size is reduced. You don't drop the size, let it grow back, and then apply the stress. You drop the size so your 1 in 100 individuals is less than 1 survivor. And then you combine multiples to enhance the effect of stress. So the combination of size reduction and increased numbers of stressors is a recipe for extinction. The population is too small to have enough individuals who can survive in a changing environment, and it crashes. So, the application to cancer therapy. And this is how I look at things from macrocosms to microcosms. And I guess it's reductionist in a way, and it gives me something to hang on to. But it's useful to get you to think about it. They don't always have to be right. It just gets you to think about a different way of looking at it. So cancer is comparable to a species population. So you reduce the cell number, surgery, to reduce the probability of adapting to therapeutic stress. And this goes back to the radiation preoperative and at the time of surgery. You apply the stress at the time you reduce the numbers. And radiation reduces the numbers. So you apply multiple stresses before the cell numbers recover. Surgical resection does not discriminate between subpopulations that can evade single stressors. So you haven't taken out a single population, you've gone across the population. And the stressors must be independent. That is, single genetic changes cannot enable adaptation to multiple stressors. So in the case of a tyrosine kinase, one specific one, if you go and combine stresses such as inhibitors of that one kinase, they're not going to be exponentially effective. They're both hitting the same thing. And the species simply has to mutate the kinase to avoid each one of them together. And it only has to have one event. So, and what I see as the application to chordoma therapy is that surgery effectively reduces the cell number. It brings it down as well as the surgeons can do it. But based on conservation biology, apply therapeutic stressors before the recovery of cell numbers. You don't let their number come back up so they have more chance to avoid the stress that you're going to apply to them. So lack of efficacy with radiation and chemotherapy suggests the addition of a therapeutic stressor that is independent in action. And immunotherapy may really provide such a function. So now I'm going to try not to bore you to death with the immune system. Okay, so it evolved to recognize the difference between self and non-self and eliminate anything foreign. And it's pretty good at that. We're all around. There are pathogens, virus, bacteria, fungi. It responds to tumor cells, and it responds to tissue and cell transplants. And one could say, well, it didn't evolve in mammals to respond to transplants. But that's the way it is now. Transplants are reality. And there are two parts of the immune system. One is innate and one is adaptive. Innate is the immediate non-specific responses to pathogens. You have a virus come in, influenza, and a bunch of cells have receptors for it. They get all torqued and make cytokines. Then there's adaptive, which is our antigen-specific responses that can develop long-term memory for accelerated recall. You saw flu last year, you still have memory cells. You see flu this year, the memory cells respond in an accelerated way. So the adaptive system maintains memory, and it does it through recognition through antigen-specific receptors. Very specific, and that's what I'm going to focus on, is adaptive immunity. B cells make antibodies. And those antibodies can see things on the cell surface, they can see things in plasma. T cells can recognize antigens a much wider range. They recognize antigens in the context of self-HLA molecules. And they've been the focus of immunotherapy. Now, HLA molecules, one complex, and Soldano Ferroni is working on, as was talked about, on loss of HLA function. It's a set of genes whose job is to present antigens to T cells. They're a complex, they form a groove, and a peptide sits in the groove, 9 amino acid, 8 amino acid peptide, and that gets presented to a T cell. And the T cell receptor sees HLA plus the peptide. So it's a very small part of the protein that the peptide came from. And that's why T cells are so specific. And peptide HLA complexes are presented by antigen-presenting cells, a whole class of different lymphoid cells. And they're professionals. That's what they do, okay? And they're good at it. And they're part of the innate system. And they prime T cell responses. And you have CD4s. I have to take you through at least some immunology, okay? CD4 helper T cells produce cytokines that help the responses of B cells and T cells. They recognize peptides presented by HLA class 2 molecules, okay? While activated CD8 positive cytotoxic T cells produce cytokines, but in the process of activation and expansion that they go through, they gain cytotoxic potential. Okay, that is, they can kill targets and do kill targets, and they do it very efficiently, but they're not great at wiping out big populations. They're good at going in and finding the single cells. So everybody now, at least in the areas that I'm interested, focuses on CD8 positive T cells. I see I'm going to be in trouble here. Okay. How tight are we?
Well, we have a few minutes for questions. So it depends on
Okay. If I confuse you thoroughly, then you won't have any questions. So. Okay, so I'll try to pick it up. So CD8 T cells, lyse target cells, they have multiple mechanisms of doing that. So there's a pretty good range of cells they can kill. And they can see, I'll try to cut to the chase on this. They see class 1 plus peptide on the surface. But those peptides are generated from inside the cell. And that is the processing machinery that gets the peptide into class 1, it goes out to the cell surface, and class 1 needs that peptide bound there to stay on the cell surface. Then a T cell can see it. So CD8 T cells can see peptides from proteins in any part of the cell. So imagine that the T cells are like your trash man coming down the street. So it stops at your house, dumps the bin, and looks through your trash, and you can see basically what you've been doing for the last week. Okay, and goes to the next person, and they just see something that they might really like you threw out something you shouldn't have. Okay, but this is the way the immune system monitors the cells. So, and I can probably cut through a lot of this and give you some more logic, but the brachyuria gene has become very prominent. It is, for immunotherapists, it is the perfect target, the absolutely perfect target, because the cancer cells can't get rid of it without dying. They can't delete it, okay? They could change the peptide, but with very low probability. So it's so required for growth and expansion that even if they delete it and the T cells can't see it, they have killed themselves. Okay, so it's seemingly the perfect target for T cells. And that's what's prompted all of the vaccine development, and there are multiple approaches to priming T cells. That's the first thing, you got to prime T cells. And there are now, as pointed out, clinical trials that are using brachyuria to stimulate with virus, with yeast. Co-stimulatory molecules are being added to improve the vaccine. And one vaccine has moved to a clinical trial. This is the point I want to get to. And that is that targets and T cells have confrontations. They are fighting each other, not, consciously, but this is what happens. So the CD8 T cells, in order for them to eliminate virus-infected or tumor cells, the targets cannot change their antigen expression. No chance of changing it, then they can wipe them out, at least within reason. But the targets don't sit back and just take the hits. Okay, influenza viruses mutate their antigens. They can't get rid of the antigen because they wouldn't grow. So what they do is they mutate the peptides. And that's what causes the changes year to year in viruses. Okay, and they have a lot of viruses, so even though it's a low probability event to mutate, they make up for it in the population size. And what you come into is this confrontation predicted by what is now known as the Red Queen hypothesis, which comes from through the looking glass, where the Red Queen has to run to stay in place. Okay, it's also referred to as a arms race. So influenza mutates. The T cell populations in individuals have to draw out new T cells to respond. Next year it mutates again, the T cells respond. And in the real world, in conservation biology, say foxes and rabbits. The rabbits learn to run faster, the foxes then run faster. Rabbits learn to corner faster, foxes learn to corner faster. So they haven't gained anything, they're still in the same place, but they've been doing a lot of work, okay? And cancer does the same thing. And if you have T cells responding to cancer, they can delete the target antigens. As I said in brachyury, they can't delete it. But if there are more target, and if there are more target peptides, then the T cells can respond to them unless they are deleted as well. But deleting multiple antigens is a low probability event. So the targets then, the tumor cells, delete HLA, which is what Soldano has found, that a number of the genes within the HLA complex that are required to get peptides on the surface are being deleted in chordoma cells. And they go, what the cancer cells aren't thinking about this, but they go to the one point that they can shut off all antigen presentation. And Soldano, I think, has proposed that because there's this selection, there must be multiple peptides. Well, it could be just brachyury. And since they can't get rid of brachyury, they get rid of what presents it. So they are now invisible to T cells. And to take the Red Queen hypothesis further, okay, which is what basically my last slides are, T cells have to be primed, they have to migrate, and then they have to do something when they get there, like kill targets. And tumors have developed ways to dodge those, okay? And T cells migrate through the body, but they need a reason to stop. And there's a classic example from Mark Davis's lab on high frequencies of melanoma-specific T cells in the circulation in patients with melanoma. They're going right by. They're in the bloodstream and they're not stopping. So tumors have developed ways to lay low. And then there are immunosuppressive tumor environments. You know that tumors infiltrate, but there are mechanisms by the tumor as well as other immune cells that reduce the ability of T cells to respond. And one of the biggest players in tumors are myeloma-derived suppressor cells, which are macrophages, antigen-presenting cells, some derived from granulocytes. And they come up in normal inflammatory responses. And they're there to bring, when the response gets done, they're there to bring it down. They bring it back down to homeostasis. But they get into tumors and they bring it down to homeostasis. They inhibit T cell function. And they do it in part through the PD-L1 ligand that was talked about earlier. It's one of their many mechanisms. And PD-L1 has been shown to be expressed by lymphocytes infiltrating chordomas. Are there populations of MDSCs in chordomas? And I think pretty soon somebody will look at that. And that's another way that the tumor has gotten around the immune system. So for me, sort of how I look at this, there is a pathway for immunotherapy. And that is activation of tumor-specific T cells, optimal vaccination for the greatest number and avidity of CD8 positive T cells, and monitoring the number of responding T cells. As you go through these immune therapies, you have to monitor how the immune system is working. Is it working? You have to know then, do the tumor cells still express their antigens? Let's say they have lost HLA. Well, why would you then vaccinate that person to an HLA-presented peptide when, even though you can make the T cells, they're never going to see a tumor cell? So you need to know what the tumors in each individual are. Now you're getting to personalized medicine. Then you need to get them to migrate, and you need to get them to sustain their functions when they do so. And so, to me, these are just my conclusions that conservation biology provides a framework for a natural selection view of the battle against cancer. The first thing you have to do is identify the characteristics and potential weaknesses on a case-by-case basis. Then reduce the tumor size and genetic diversity and apply selective pressure to the remaining tumor cells, specific for the individual case, and multiple and simultaneous stressors to increase probability of extinction. So that's where I'd like to stop.
Thanks, Peter. Questions for Dr. Wettstein?
A ketogenic diet has been used effectively against some glioblastomas and also has been used in treating seizure disorders. And the hypothesis is that by removing carbohydrates from the diet and focusing on ketones as an energy source, you reduce the energy to the tumor cells. And I just wondered if that has been looked at vis-a-vis chordoma. I know the metabolism of chordoma is different than what we would consider as normal cancer cells, if there is such a thing. But I just wondered if there's been any looking at it.
I'll leave that with you.
Never mind. So I think the short answer is we don't know. But it's definitely something that a topic of conversation has come up at past research conferences and something that investigators in the field are aware of and are looking into.
Thank you. We have a question here. So,
I just had a simple question. Do you know, is insurance typically covering immunotherapy at this point?
I have a PhD after my name. I stay away from those guys who are making those decisions, but you get to a good point. In order to make this work, there are a lot of steps. As I said, you don't want to vaccinate somebody who can't respond. So you have to pre-test, you have to vaccinate, you have to monitor. And somebody's going to have to pick up all of those. And if you don't monitor, and the studies that are coming out, they monitor T cell frequencies. That's the sort of thing that has to be done so you know how effective your treatment is. And once you get into multiple treatments, then you have to monitor multiple treatments. So I don't know. I'm sure they will resist it, however.
Just that really quickly. So based on our experience and really Shannon's experience with patients who have whose physicians have recommended a checkpoint inhibitor, for example. Shannon can correct me if I'm wrong, but I think the experience has been mixed. Some people have had luck, some people haven't had luck. That's one of the advantages of considering on a clinical trial is that the actual cost of the drug is donated by the company, so patients don't incur that cost. And there will be a clinical trial starting at Sloan Kettering and Johns Hopkins testing a checkpoint inhibitor in combination with radiation later this year, early next year.
Thank you.
Sure.
Yeah, fascinating. So many different pieces to put together when you're thinking about an ecosystem and the society and then relating it to a cancer. And I was thinking about, I had wide margin surgery. If you have a species only in the state of Kansas and you destroy all of Kansas, then it seems to me that the ecosystem doesn't play a role in the rest of the states. Now, people in Kansas, please don't jump all over me.
I
But
was going to say,
that's one size. That's one side of your theory. And the other part is if it's always possible that the species is not always in Kansas, seems always to be in Kansas, but not always in Kansas, that it will hide and come back another time. So, if a tumor always has the possibility of coming back at another time, even with any kind of vaccine or therapy, will we ever solve the problem of chordoma?
well, I think it depends on if there are metastases or recurrences, you have to have a good idea, did it come from Kansas first? And you should have a sample of Kansas as well as a sample of originally the other 49 states. So you ask, are the survivors clearly from one or the other? Okay, the recurrences, and you treat them accordingly to, and I am just so avoiding political, so I'm trying to frame my answer properly, is you want to know where they come from and what the starting genetic diversity was before you make decisions on how to treat them. Does that make sense?
Oh yeah, I just said here.
So in other words, you treat a Kansan entirely differently than somebody from New York. Okay.
But a follow-up is the fact that we sit here, some of us, believing that the wide margin surgery was satisfactory. And yet we know that every so often, someone who had wide margin surgery has a reoccurrence. So we are in a situation both with the Kansan and with your idea that you never are going to solve the problem.
Oh, I think you can solve the problem. You can solve the problem, but as I was putting this together, I was thinking, boy, this is really pessimistic. But I was trying to give you a realistic view of what the interplay is. That there is a response, then there's another response. But what you have to do is identify the stresses that are the most destructive. So you drive the probability way down.
It's like the old thing in college. If a person goes halfway to the frog, each time the frog takes another step, you never get there.
That's right. So you probably have to do something to the frog. Just shoot the frog.
That's the solution. It's the frog.
Shoot the frog.
We're missing it all these years.
Yeah, and now everybody can carry, so probably you're well equipped.
The question about diet was interesting in the sense it's sort of a timely topic. And there was a seminar given, Science had sponsored it on Thursday, and you can access it on the web. And it's called immunometabolism. And essentially what they were looking at is the fact that tumor cells preferentially metabolize sugar, as you say. It's called the Warburg hypothesis. It's the basis for the PET scan because you give a radioactive sugar. Well, it turns out when immune cells get activated, they alter their metabolism, and sometimes they shift as well to anaerobic metabolism, and some of the intermediate metabolites affect the immune response. So this seminar was from someone at the Max Planck Institute in Germany, and then from an investigator in Dublin, and they were talking about the interactions between the immune system and the metabolism of the tumor and the T cells. It's fascinating data, and so this is just sort of emerging as something that we have. It was something I would have never thought of. It was interesting.
Well, the myeloma-derived suppressor cells, one of, or a multiple way of their inhibiting T cells is that they have arginase 1, so they convert arginine, then they convert tryptophan, that T cells also need, and there's, I think, cysteine. And so what these suppressor cells do is, and it's not really specific, they just sort of destroy those amino acids in the neighborhood. And if the tumor cells don't need those amino acids, that is, they can, make them themselves, they continue on, but the T cells are inhibited. So again, it's a way that the tumor cells have adapted to fight back. And all they're doing is making use of what these suppressor cells normally do. And these suppressor cells keep inflammatory responses from going wild. Once the virus is gone, once your flu infection is treated, they knock down the T cell populations. Well, unfortunately, they're being co-opted to do the same thing. So there are a lot of ways to do things, and some of them work in culture, some of them work in animals, some don't work in animals. And the preclinical is really necessary. But the important thing about immunotherapy is you go look for an antigen they can't do without and that other cells in the body don't have. And brachyuria is pretty good.